So this slide is mainly telling about the applications of this sphaeroplast and protoplast. The membrane balls that are protoplast or sphaeroplast, which can be induced to fuse. They are capable of fusing with similar structures. That means they are capable of fusing with more easily with similar structures, also with the other types of structures such as eukaryotic cells. They are capable of fusing with similar structures as well as with eukaryotic cells. And this facilitates what? The transfer of genetic material between these two cells. And also another important application is that the sequential manufacture of this sphaeroplast and protoplast allows the selective release of contents of the periplasm. And their application comes that this approach, that means the selective release of contents into the periplasm, this approach has been popular in the identification of what? The components of the periplasm, because there is a selective release of components onto the periplasm, and also we can also helps in finding what helps, and also it helps in the localization of the proteins to one or other of the gram-negative membrane. For example, if certain proteins are present in sphaeroplast population, but when that proteins are absent from a protoplast population, then we can assume that the protein is absent in the protoplast population but present in the sphaeroplast, which means it can be either located in the outer membrane, because protoplast doesn't have any outer membrane because it is derived from gram-positive bacteria. But if the proteins are present in the sphaeroplast population, so it might be present in the outer membrane because it has been developed from the gram-negative bacteria. So these are the applications. Now coming is the detailed applications of sphaeroplast and protoplast. It can be used in DNA transfection. That means that this point I have discussed. The laws of the cell wall means that these cells can be induced to fuse with other cell types. This can be useful for the transfection of DNA into the animal cells. This I have discussed in the previous slide. And also it is useful in plant biology, allowing what? The fusion of the protoplast from the different species, and it results in the formation of somatic hybrids. Plant protoplast can also be used to recreate an entire plant from a single cell by forming a callus. Next is patch clamp analysis. So, while preventing the cell division in the sphaeroplast, it may result in the formation of a giant sphaeroplast, and this can be used in patch clamp analysis. This can be used in patch clamp analysis, and this is useful to characterize bacterial ion channels. And this method monitors the flow of the current through this channel, and a singular bacterium is too small for this assay. That's why we are using giant spheroids, because they are large enough to allow the patch clamp recording to record or monitor the flow of current through this ion channel. So using this sphaeroplast we can perform patch clamp analysis. This is another application of sphaeroplast. Next, we can use this sphaeroplast for characterizing antibiotics, which means these giant sphaeroplasts are grown in the presence of an antibiotic called cephalexin, which prevents the cell from splitting and dividing. And this may result in forming a snakes with single membrane and cytoplasm. So, it may result in the formation of a single membrane and cytoplasm. These snakes can then have their cell walls removed and the resultant spheroid can be used in the patch clamp analysis. So this can be later used in patch clamp analysis. So mainly for the DNA transfection, patch clamp analysis, characterizing antibiotics, we are using this sphaeroplasts as well as protoplasts. And we can also detect the amount of certain types of protein, that is flow of certain substance or proteins within the periplasm, that can also be detected. So these are all about sphaeroplast and protoplast. Thank you.